Right, hello everybody. Thank you for joining us. I'm hoping we can shed a bit of light onto this topic, transport in plants, which is often seen as a more challenging topic because of the sheer number of new terms that you have to get used to during the course of the topic. So we'll see what we can do. The plan for today is that we're going to look at some of the basics of vascular tissue and look in a bit of detail at the structure of and function of xylem. If we get time today, we'll move on to phloem. We're going to look at focus in a bit more detail at xylem and the pathways of water and minerals, the pathways that they follow to go from the soil into the xylem vessels. If we get time, we'll move on to transpiration and the effects that I have on the plant, and indeed the adaptations of different plants according to their environment, according to the nature of their environment that pertain to the water availability. And then we will move on to the mass flow hypothesis. Now, there's a number of these sessions running, and I will endeavour to cover as much of this today as I can, but failing that, the intention will be to use a later session to finish off anything that we don't cover today. So hopefully over the course of these sessions we will cover the whole section on transport in plants, and we'll see how we get on. So let's first of all have a little overview of what we mean when we're talking about transport in plants. So first thing, we need to look at what we mean by vascular tissue. Now, that's quite a broad term and simply means any tissue which transports nutrients and water throughout the plant. And actually, it's rare in any exam questions for them to refer to vascular tissue. Really, it's an umbrella term under which we have the subcategories of xylem, which you hopefully will remember from GCSE, but it functions to transport water and minerals from the roots up the plant, up the stem towards the leaves. And then we have phloem, and phloem kind of works in the opposite direction, in the sense that it transports the products of photosynthesis. So think about those leaves are the sites of photosynthesis. So at the leaves, we're going to make glucose, but we'll also make other things from glucose, such as amino acids, and those things will need to be transported from the leaves to other parts of the plant, which are usually the growing regions more than anything else, which would be the shoot tips and the root tips. So we'll need to transport those products. Usually we talk about the transport of sugars, but actually it's other products of photosynthesis too. Now on the right hand side, you see a picture here of the distribution of vascular tissue, and that's something we're going to focus on a little bit more detail. But let's try this first of all. So what we just covered there is the real essentials. I'm going to just give you a couple of moments to look at what I've got on there and see if you can think about what words are missing from there. Failing that, you can always pause the video and take a few moments to have a look at that yourself. Okay, so number one there, it's the vascular tissue, which transports nutrients and water throughout the plant. And the two main components of vascular tissue I mentioned are xylem, which is all about transporting water it goes from the roots to the leaves, and the phloem, which transports the products of photosynthesis from the leaves to other parts of the plants. Now, it's worth mentioning that this is something I'm going to try and do repeatedly, where I might introduce an idea and then ask you to do a little bit of recall, or give you the opportunity to do a little bit of recall, just to try and make this less passive and get you more actively involved in trying to recall some of these things. And that just means it's more likely to stay with you. So let's focus a little bit more on the xylem. Okay, so now the xylem, it would be lovely if the xylem was quite simple. It was just a straightforward tube which transported water. But unfortunately, we talk about xylem tissue, meaning we're talking about more than one cell being present. And those cells tend to have somewhat different functions. So we've got two components of xylem tissue which are responsible for transporting water. So we have xylem vessels, which are continuous tubes, which are, if you like, the better adapted tube for transporting water. What you find with xylem tubes is that they used to be cells stacked end on end, and then those cells lost their cytoplasm and lost their end walls, and then just functioned as a continuous tube running through the xylem tissue or running through the plant. Now, the alternative is what we call tracheids. So you can see the xylem vessels on the right-hand side here, and you can see you've just got these continuous tubes, and it shows there an end wall, and it says with a single large perforation, so just one big hole really. And so water and nutrients, or water and minerals, I should say, are able to flow with little restriction as they pass through xylem vessels. Now, tracheids are a little bit different. Tracheids also are responsible just for the movement of that water and minerals, but they do still have their end walls intact. Now, that's obviously going to slow down the movement, but they do allow movement of water and minerals through because they have these little gaps or perforations in their end walls to allow water to move through those end walls into the next tracheid. You'd also notice the tracheids are sloped or tapered, as those end walls are tapered. And it's thought really that the xylem vessels, like I think I said, are the sort of more advanced means of transporting the water and the minerals. The tracheids you're more likely to see in sort of more primitive plants, and you're perhaps more likely to see them in shoots or root tips, shoot tips or root tips, and that kind of works out nicely given that you've got these tapered end. It kind of looks more like it's going to be moving into the shape of a shoot. So both xylem vessels and tracheids are responsible for transporting water, which we think is the be all or end all of the xylem. And yes, for all intents and purposes, that's all we're interested in. But there are the other components to xylem tissue. So you have these things called fiber cells. Now, fiber cells are shown in the middle here. I'll try and make a little bit more sense of this diagram shortly, but the fiber cells are shown in the middle, and they look like sort of normal cells, if you like. They've got very thick walls, you notice on there. Their function is there for support, whilst the other cells that you can see, which do seem more like normal cells, because we have a nucleus and a vacuole, and well, cytoplasm we can see there too, but it certainly looks more like a standard plant cell. We refer to this as parenchyma. So parenchyma is just, well, like I said, it looks like normal cells, and in the context of plants, we really regard parenchyma as being packing tissue. So it just sort of fills out space. So you can think of it as not having much of a function other than taking up space in between sort of the business areas of the plant. So the fiber cells are all about support, and the parenchyma is all about forming this sort of packing tissue, and we'll see a lot more parenchyma shortly. And there's not much going on with parenchyma. We don't really need to talk about it a great deal, but you will see it, and I can just say, look, there's some parenchyma. Yeah, it's not really important because it doesn't really have a major function other than packing out some tissue. Some other important points. Okay, so first one is lignin, and this, now I can talk a little bit more about this diagram. So lignin is a waterproof substance which deposits itself in the lining of the cell walls of the xylem vessels and the tracheids and the fiber cells. Now, when it deposits itself in that cell wall, you'll see that it starts the first level of deposition is this sort of spiral arrangement of lignin. So when a cell first starts to become lignified, stage one is this sort of spiral arrangement that we see in this this third cell here. Then on the far right hand side, we're seeing the next stage, which is like the double helix arrangement or double spiral arrangement. Looks like a DNA molecule there, and then those start to fuse together a little bit more, and we end up with this arrangement we see in this one from the left hand side here, this one where you can see they've started to join up, and finally a fully mature xylem vessel looks like the one on the left here, because we've got it's almost completely lignified, but we've got some pits in there, some pores, which do allow movement of water from one xylem vessel to the next. You can see that the fiber cells are completely lignified, and the tracheids and the xylem vessels would be more and more lignified the older that they would have become. The parenchyma, you don't see any red on there. Parenchyma is not lignified at all. Whenever you see lignification, it's telling you straight away that that cell is dead, which tells you that the xylem vessels, the tracheids, the fiber cells are all dead cells. Their function is, well, fiber cells is simply for support. The xylem vessels and the tracheids, obviously their primary function is the transport of water and minerals, but they do have an additional function of adding support because of that lignin that strengthens those structures. But the parenchyma, you can see, doesn't have any lignin, so it is the only cell in xylem tissue which is alive. So having said all that, this is always a good activity that I'm going to show you now, but I always think it's a good idea whenever you get something as complex as the structure of xylem with lots of stuff going on. It's always a good idea to try and represent that yourselves, to take a bit of time to do it yourself and think, right, what's going on? You can read, but you struggle a little bit when you read to take everything in so that it's memorable, to represent it in your way. So I always encourage students to draw what they've seen. We don't know with everything, but for something like this, where there's not a huge amount, it's often quite difficult to sort of see the wood from the trees, so to speak, is to draw what's going on. And this is an example I've from a previous pupil showing a xylem tissue where we've got quite nicely shown parenchyma on the left-hand side there. You see they've labeled it as parenchyma. I've got a mature xylem vessel next to that. You've got a little cutaway there so we can see the pathway of water moving up through the parenchyma. Sorry, up through the xylem vessel. And then I got a slightly less mature xylem vessel here because we don't get that lignin all the way through. You got pits linking those xylem vessels together. I've got some fiber cells. They've got a degree of that green color showing that they're lignified too. And then I got tracheids. And the tracheids have got their end walls still intact, but with some pores or pits, perforations that allows water to pass through those end walls. You see it's a tapered end. You can see it's got a hole there or a pore there for water to pass through. Now, one of the it's nice that you're able to see a version that another student has done, but I would say that it's important that taking time to actually represent this yourselves replaces that section in your revision, which would be like making your own notes, where you might write, I'm going to show this how I see this, get everything the way you want to see this, and that time you take puts you in a good position to then go forward and move on a stretch. And to that extent, I'm going to do the same as we just did here, given everything I'm going to do the same as I just did previously, given everything I've just said. I'm going to see how you get on with answering these questions and filling in these blanks. So you can pause the video here and take your time and try to remember what things should appear here. Right, let's have a look. So xylem made up of 4 different types of cells. So we've got the vessels, or the xylem vessels. Here the cells found in a long line, but the end walls of the cells are lost, so it forms a continuous tube. Then we have tracheids, also conduct water. I can add on here that minerals too, but we'll take it as a given now. But they're not as well as adapted as vessels for this function. They have tapered ends that fit together, and then they have pits or pores to allow the transfer of water between these cells. Then we have fiber cells, which are dead. We'll talk about lignification in a moment, that have no role in water transport, but function and support. And we have xylem parenchyma, which acts as this packing tissue. Vessels and tracheids are dead cells, which together form a system of tubes through which water and minerals can travel. These dead cells have no cytoplasm, so there's nothing to impede the flow of water. Vessels and tracheids and fiber cells have lignin deposited in their cell walls, making the wall impermeable to water, so water can't pass through it, and to solutes or minerals, whilst also providing mechanical strength and a support to the plant. Now, something I encourage, so when you think about if I try and help a little bit with preparation for when it comes to tests or exams, as you sit there right now and you're listening, that might be useful for you, that you can maybe get some clarification, excuse me, on stuff that hopefully you've heard of before, but you have to go that next step. And so you have to get more involved in that. So you sit there and you're passive. In order to go a little step further, you've got to start doing something. And that's a little bit like why I start with this. And often I do this in my lessons where I will have a fill in the blanks exercise and ask people to write them down. And I do that on whiteboards. And then what I do quite often is say, look what you've got here written down. Look at the right answers. So we go through the right answers, and you see on the right hand side now are all the answers taken from that previous slide. And then I say, okay, well, let's see if you could go to the next step now. You've got those keywords that are in there. What if I ask you to describe the structure and the function of the xylem using those words? And if I was to then take that away, the previous slide, and say, okay, how would you go about doing that? And this is a big step forward now from filling in the blanks, which is quite a low level activity, to now working on your understanding. So I'm big believer that when you start to explain to other people, or just explain to a empty room, or to a wall, if you like, that's when you start realizing what you do when you don't know, or what you do when you don't understand, because you aren't able to piece together, use the linking words to put together coherent sentences. And so you can use other people for this, but you tend to know yourself when you're realizing you can't describe something appropriately. So it's always a good activity to take the words or keywords from a particular topic and then try and explain what's going on, in this case with the structure and function of xylem, just using those words, okay, or using those words as waypoints, okay, as guides. Okay, let's have a look at how xylem is distributed throughout the plant. So we talked there about xylem being having the lignin within the cell walls, adds structure and strength, a support to the cell, and therefore xylem does function throughout the plant, not just in transport of water and minerals, but conferring some strength and rigidity to the plant. And therefore, as a result, the way it's distributed through the plant is indicative, or is related to the needs of those different parts of the plant. So I showed you this picture earlier, but let's have a look at a little bit more detail of what's going on. So look in the root. In the root, xylem has this central arrangement. So the xylem here, which is shown in red, you can see that the xylem is this sort of star shape, but it's right in the middle of the root, and is centralized. You can see the phloem on the outside, but we're not really interested in the phloem right now. You can see these little dots of phloem. We're not really interested in the phloem because the distribution of the xylem has a significance in conferring strength and support to the plant. Phloem doesn't do that. So while xylem has a role in transport of water, we're interested in where xylem exists in different parts of the plant because of what it confers to that plant. So you can see the root has a central arrangement, and the reason it has that is because it resists that vertical stress, that being pulled out. So it helps to anchor the plant. Now, when you move up to the stem, it's got an arrangement which we call a peripheral arrangement. There's a peripheral vascular bundle. You can see that you've got a vascular bundle consists of both xylem and phloem. But again, we'll focus on the xylem. You can see that it's a peripheral arrangement. It's around the outside of the stem. And the reason for that is by having that arrangement, it makes the stem more flexible, more resistant to bending. And that's obviously a stress that a stem would have to deal with, as opposed to a root. A root doesn't have the issue of it doesn't need to be flexible. It doesn't need to resist bending. Its job is to anchor the plant. Whereas a stem, its job is not to anchor the plant. It doesn't need to resist vertical stresses, certainly not in the same way. It needs to resist the bending that we'll experience in the wind. And then the leaves have got a different arrangement. Again, you find xylem within the midrib of the leaf. Okay, so this central vein that runs down the midrib of the leaf. And then when this midrib branches off, we end up with this network of veins. And what that means is by having the xylem in that arrangement, it says it's flexible, yes, but it also resists tearing. And you've probably all experienced this when you try and tear a leaf. If you're trying to tear it down the line, it's sort of showing on the image here. What would happen is the first bit would tear without a problem, but it would then meet a vein, and then it would tear along that vein rather than tearing across that white line shown on the image there. It would tear along the vein, and it would meet the midrib, and it would then stop tearing, because it's actually very hard to break through that xylem that runs through the midrib, or indeed the xylem that runs through the vein. So it resists tearing. So look at that. Let's take that a little bit further and look at what we would see in a root if we look through a microscope. So I mentioned previously about the problem of this topic is there are so many new terms here. Something I try and do, sometimes I do this at the start of teaching, sometimes I do it at the end of the teaching. I don't know which one is best, but I have a list of all the terms, and I have a list of all the definitions. And I said, this is something that we have to nail down. We have to learn these. And there's various applications for doing that. I mean, you can do it on flashcards or whatever it may be, but there's two advantages to that. The first one is when it comes to an exam, when learning definitions, particularly for this topic, when it comes to an exam, having in your locker the opportunity or the ability to just reproduce a definition rather than have to think about it too much, but because you've learned it and you've learned it by rote, often picks up marks that you didn't really expect to get, because it's not done with a huge amount of consideration of the question. You say, well, if they ask about xylem in the root, you can be dropping in terms like the steel, the endodermis, the Casparian strip. You can talk about these things, you can drop them in without necessarily thinking that a key part of your answer, but you pick up marks in that way. And the other reason, those terms I'll come on to in a moment, the other reason is because when you're then moving on, so this is the advantage of learning these terms at the start of the unit, is that when you're then working through that unit and these terms are being used all the time, you don't have to keep saying to yourself, oh, what does that mean again? Or what was that about again? You have those terms at your you're at liberty to use those terms and understand those terms very quickly. So let's have a look a little bit at what's going on in this root then. So we've got here an outer, so we've got really two parts really to this to the root. We've got the cortex, which is everything from the very edge of the root up to this central part, which is called the stele. So we've got an outer layer of cells, which is everything here, that is full of parenchyma, packing tissue. Okay, so the cortex is full of packing tissue, parenchyma. And then when we zoom in on the stele, this is the business end really. So this is what I say about everything that appears in the cortex. We're not all that interested in. It's not a huge amount going on there. We'll look at what does happen there shortly, but not a huge amount going on there. This is where the important stuff is within the stele. So the stele is recognized by, you can see it's a circular arrangement within the middle. Its outer layer of cells, the outer layer of the stele is called the endodermis. So think of dermis as being a skin, and endo means within, is from the Greek within. So endo is like the skin within, endodermis is the skin within. So the outer layer that's within. So you've got endodermis here, and then we've got our xylem here. And the xylem you can recognize, first of all, it's a star shape, the xylem vessels. Okay, or tracheids as it may be, but usually it's xylem vessels, and they've got thick walls, okay, because they're lignified. It's very notable. If we then go on to the stem, so this is the peripheral arrangement of vascular bundles in the stem. We can see we've got this arrangement. You see these darker sections all the way around the outside, and that consists of a xylem on the inside and phloem then on the outside. Okay, so you've got xylem and phloem there in each of these little vascular bundles, which appears slightly darker all the way around the outside, a peripheral arrangement. Now, another one I ask pupils to do, let's draw what you've just seen there. So this is an activity. I'm going to show you this again from a previous pupil. So they've shown up this stem, they've got a peripheral arrangement there, and you can see they've identified there fiber cells and phloem and xylem. So you see xylem on the inside, but the key thing is that it's showing it on the outside. And why we do that is because those plants get flexible support and resists bending. And then in the root, they've got this central arrangement, this star shape within the middle, and it resists vertical stresses. It anchors the plant. Okay, and they've identified this as being this area in the middle of the stele. Okay, let's try this one more time. So look what we've got here. Again, you can pause the video as you see fit and then have a go at this yourselves. So distribution of xylem varies depending upon the location in the plant. In the roots, the xylem has central arrangement to resist vertical stresses and help anchor the plant. In the stem, the xylem exists in several peripheral vascular bundles towards the outer edge of the stem. That's what peripheral means, as this gives the stem flexibility and resistance to bending. In the leaves, xylem is found in the midrib of the leaves and within the network of veins, makes the leaf flexible and resistant to tearing. If I move on, just like we did before, be ready to do this. This is a good opportunity for you to now take those keywords, take away that support, and then try and explain or describe to someone else, describe and explain what's going on in the arrangement of xylem, what the distribution of xylem and the why. So I think it's the last time I'm going to show you this opportunity of taking those keywords, but note that this is something you can always do, albeit using this presentation that I've used, or this this recording today, or just picking out keywords from a topic and then taking them and trying to explain to someone else. Nothing prepares you better for learning and recall than actually explaining, I say to someone else, explaining out loud. Okay, it doesn't have to be anyone there. You know when you start to lose that coherence. Little recap. Just give you a couple of minutes, or you can pause again. I'll give you a few seconds now to have a look at some of these. So you asked there now, what components of xylem tissue are involved in the transport of water? So we're not going to be talking about fiber cells, are we? We're not going to be talking about parenchyma. We're only interested in xylem vessels and tracheids. What do we call the arrangement of xylem tissue in the stem? It's not a central arrangement, that's the root, and it's not in the midrib in the veins. It's the peripheral arrangement. The name of the substance, the waterproof substance deposited in some of the cells found within xylem tissue, that's going to be lignin. Where is xylem found within leaves and why? It's in the midrib and veins, and why it's flexible and resists tearing. And the general name for tissue which transports water and nutrients throughout the plant is just vascular tissue. The next stage, and this really now is where we move to the business end of transporting plants, about how we get water from the soil into the root, the transport of water into the root. So everything we've covered so far was necessary. We need to look at the structure of xylem. We need to look at the distribution of xylem, but really we need to look at sort of its functioning and how we actually go about getting that water from the soil into the xylem itself. So let's go back to this diagram here. Well, look, if we look at this first one, we're seeing the whole of the root, cross section of the root. Or transverse section, this as it shows here, transverse section of a mature root. Water's got to get from the outside of that root, it's got to get through the cortex, meaning through all the parenchyma, and then it's got to get into the stele and into the xylem. Okay, I show it on this side here. So water's going to come all the way through the parenchyma, and then it's going to cross the endodermis, the endodermal layer, and then get its way all the way into the xylem vessel there. That's what water needs to do. Now, know from GCSE that plants have root hair cells. So those root hair cells are found in the epidermis, the outer layer of the root, and they function to increase the surface area for the absorption of water and indeed minerals. Now, it's worth keeping that in mind. You do get asked questions on that. It's a GCSE concept that we first come across that, but we do need to appreciate that that's still relevant here, though we tend to overlook it perhaps to our peril really, because questions do get asked, but we tend to focus more on what happens once water gets towards this central stele area. But let's not forget that. Right, so this is an activity, but I'm going to use this activity to sort of demonstrate, to model what's going on here. So this is an activity I ask pupils to do. I give them this diagram and I say, can you draw and annotate cells around the Casparian strip, including the pericycle? The pericycle, again, this is one of those terms that crops up here in transporting plants. The pericycle is the cells just inside the endodermis. So these cells here correspond to the pericycle. So it says, draw and annotate cells around the Casparian strip, which really we're talking about the endodermis, including the pericycle and the central xylem vessel, and show the pathways which water can take through the root of the plant ending in xylem tissue. So there are three pathways that water can take. Okay, the first pathway that water can take is what we call the symplast pathway. In order for water to pass through the symplast pathway, it needs to get into the cells and then will pass from cell to cell through the cytoplasm and via plasmodesmata. Plasmodesmata is a topic you would have, or a term you would have come across in 1.2, cell structure and organization. It's easily overlooked, but it's something that is an essential component to transport of water and minerals into the xylem, because it is these bridges, these pathways where there's no cell wall effectively connecting the cytoplasm of one cell with the cytoplasm of the next cell. That little passage there is called a plasmodesmata, they're called plasmodesmata. So the first pathway we're going to refer to here is the symplast pathway, and I'm going to show you the symplast pathway. It's very straightforward. So water gets into the outer cell. Now, how it does that, that would be enhanced by a root hair cell, of course, moves in by osmosis. We'll actually look a little bit more at the role of minerals in doing that shortly, but this is a very straightforward pathway for water to move, because it just goes through the cytoplasm and plasmodesmata. Do not just say cytoplasm. Okay, you must refer to the fact that the water is moving from the cytoplasm via plasmodesmata all the way into the xylem, but you must refer to plasmodesmata there. Symplast pathway. In fact, the term sim is from the Greek or Latin for same, like sympathy, to feel the same, and plast refers to the cell. So symplast is like refers to water moving in the same cell, because these cells are joined via plasmodesmata. It's like they're one big cell, because they're effectively all joined together with continuous cytoplasm. Okay, the next one I'm going to refer to is the vacuolar pathway. Now, often we refer to the vacuolar pathway as transport from water from vacuole to vacuole. It doesn't really work like that. It's often stated in textbook that it's movement of water from vacuole to vacuole, but really the vacuolar pathway is just an extension of the symplast pathway. So you can see that water moves via the cytoplasm. It may well move into the vacuole and come out of the vacuole of a cell, and then back into the symplast pathway, then into a vacuole and out of the vacuole, and back into the symplast pathway. So the vacuolar pathway is really just the symplast pathway with movement in and out of vacuoles added on. I'll come back to the vacuolar pathway shortly, and it's sort of its lack of importance really. But first thing I want to do is talk about the apoplast pathway. And the apoplast pathway is the movement of water through cell walls. Now, this takes a little bit of explaining. Cell walls do not convey any restriction to water at all. Water can freely pass through cell walls. There's no issue with it at all. In fact, water moves primarily through cell walls, meaning that's the main route or the fastest route for water to get from the soil into the xylem vessel. So the fastest route for water to take would be the apoplast pathway through the cell walls. So if I show you that happening here, there it goes. Water's moving through the gaps that appear effectively between cell membranes, okay, of adjacent cells, because those cell walls effectively fill in a gap between cell membranes. However, and this is sort of the most troublesome part of the water transport into xylem. However, when they reach the endodermis, okay, so the endodermis, if you remember, is this outer layer of cells of the steel. They come across this thing denoted in blue here, which is called the Casparian strip. Now, the Casparian strip is a substance called suberin, which is deposited in the cell walls of all those cells which are in the endodermis. And suberin is a waterproof substance, which if you look at this diagram over here, you can see if water tries to move via the cell wall, it will hit the Casparian strip and will not be able to move any further via the apoplast pathway. Therefore, at that point, water has got to find an alternative route into the xylem. And indeed, once water passes via the apoplast pathway all the way to the Casparian strip, it then has no choice. When it hits the endodermis, it reaches that Casparian strip, it then has to enter the symplast pathway. Now from there, actually, look, it can, once it's now entered the cytoplasm, it could leave the cytoplasm, leave the cell membrane, go pass out of the cell membrane, and then go via the apoplast pathway again. That's possible, but it's just as likely it will just stay in the symplast pathway once it has entered the cell. The key thing is that water does not have a free passage all the way to the xylem from the soil. At some point, it's going to have to cross a cell membrane, either right at the start when it enters the symplast pathway in a root hair cell, or it can move via cell walls, the apoplast pathway, but once it hits the Casparian strip, it will have to go via the symplast pathway for at least some of the time in its movement into the xylem. Now, I said I'd go back to the vacuolar pathway. It's worth mentioning that actually the vacuolar pathway is very rarely asked about, or put it in a different way, it is not uncommon for questions to ask for the two main pathways by which water enters the xylem from the soil, and they will always be the symplast and the apoplast pathway. And the reason for that is because I said the vacuolar pathway is sort of an extension of the symplast pathway. So you wouldn't use those two pathways as just say, oh, it's the symplast and the vacuolar. You wouldn't say those. It's the symplast and the apoplast. Those are the pathways, the two first ones you'd refer to. It's worth mentioning here, I'm going to show you this, and then we're going to try and do a little bit of looking at some questions on this as well. The role of minerals here. So this is a similar idea. What about the role of minerals? So you can see I've drawn on a root hair cell here. Minerals do have a role in transporting water into the plant, because actually minerals get actively transported from that soil water, that in the soil outside, they get actively transported into the those root hair cells. Once they've been actively transported into the root hair cells, what they will then do is lower the water potential, and that will then cause water to move in. So the minerals, when they're actively transported in, lower the water potential of the cell, and so they draw water in from the soil by osmosis. And then as water moves in, it's now in the symplast pathway, and it will move all the way quite happily, just like I've just shown. It's possible that it could still, of course, we know it could move via the apoplast pathway. We have the same problem, but then it will enter the symplast pathway, and I'm shown differently here. It can now move all the way in via the symplast pathway from there. So all I'm showing on this with this activity is that the additional role, or the role of minerals in the transporter of water, the transport of water. You can see that, of course, the minerals need to be transported too. So minerals are perfectly capable of moving via the apoplast pathway, but once they get to the Casparian strip, they will then have to be actively transported into the cytoplasm and then move via the symplast pathway from there. Now then, there we are. I've shown that it can move via the apoplast pathway too, once they've got inside the endodermis. So again, this isn't something that I expect us to spend a huge amount of time on here, but this is another opportunity whereby you could do a little fill in the blanks exercise, take those keywords, and this is one place, is a one-stop shop for really the key features that you need to know about transport of water into the plant. So I'll just quickly run through this. So root hair cells have a large surface area to aid with the absorption of water. It's a key fact, we need that. Water can move through cell walls, across the cortex of the root via the apoplast pathway. Water can also move across the plasma membrane by osmosis, but once within the cell, the water moves from cytoplasm from one cell to the next via plasmodesmata, and that's referred to as the symplast pathway. So I've added on that water can also move through cytoplasm and vacuoles via the vacuolar pathway, but I'm only giving that one sentence because it's far less important. Within the walls of the endodermal cells, key stuff here is the Casparian strip. Strip is made of suberin, key term there, a waterproof substance, and it functions to stop water moving via the apoplast pathway and then force it to move via the symplast pathway. Ions are absorbed into the root hair cells by active transport, so we need to keep that in mind as well. As a result, they lower the water potential in root hair cells, and that means they're going to draw more water into these cells by osmosis. Ions, which will always be dissolved in water, may also travel along the apoplast route. They can get there, but only until they reach the Casparian strip in the endodermis. At this point, the ions must be absorbed into the cytoplasm by active transport, and as the ions travel into the xylem, they lower the water potential within the xylem, and that draws water in that way. I have not now put together all those terms and asked you to do the same, like explain that to someone else, but I've given that idea now. It's something you can do. Let's finish this section now on a question that you might be expected to answer. So again, this is an opportunity for you to pause and come up with your own attempts at this, but for now I'll work through it with you. So explain how a root hair cell is adapted to its function. Kind of a GCSE idea. You can see it's one mark. You've got 3 different marking points you could refer to there. It's got a large surface area, which is the most likely thing you'd probably refer to there, to absorb water, to absorb minerals or ions. The fact that it's involved in active transport suggests that it's going to need mitochondria. So you got lots of mitochondria there. And you could also have said that it's got a thin cell wall for a short diffusion pathway. It's a 1 mark question. You only need to say one of those. Always with past paper questions, remember why you do them. You do them because you're trying to learn for the real thing. So you're not really interested in what you get right. You're more interested in what you get what you get wrong, or what else was on the mark team that you didn't say, and just taking that on board to see if that's something you want to take, you want to learn for the future. Now the next question is more the business end really. There was only 2 marks. It says water can travel across the route via the apoplast and symplast pathways. Describe the difference between these 2 pathways. That's a very nice question, really. In fact, already they're zoning in on apoplast and symplast. Notice no reference to the vacuola. Describe the difference. Very nice. I don't think you get an easier 2 marks on this. The apoplast pathway is all about travel through the cell walls, and the symplast pathway is through the cytoplasm and plasmodesmata. Note the and is underlined. It's telling you you cannot just say cytoplasm, you must say and plasmodesmata as well. Okay, I'm going to stop at that. I think that's a good place to end today, and that means that next time that we look at the transport in plants, which will be in 2 weeks' time, we can move on to the transpiration stream and looking at the role of transpiration and those other things I identified on this introductory section, where we're going to look at transpiration, adaptations of plants to their environment, and hopefully we look at the structure of phloem and the mass flow hypothesis. Okay.